Okay, so we just added the media, the trypsin to the plate,
Stop.
and
Don't do it.
so we just added the media and the trypsin to the plate, and like I said, you tilt the plate a little bit so it collects on the bottom. You can put head tip on, suck some up, squirt it around the plate, suck some up again, keep squirting it, do that 10 times. When you're done, all the liquid should collect at the bottom here. What you're going to do is, you're going to have a conical tube, preferably a 15 mil conical tube. You're going to extract all the liquid and put it in the conical tube. Include the bubbles. There will be a lot of bubbles around. You actually want bubbles because some of the bubbles might contain some cells, and you want to salvage any cells if you can. So you're going to transfer everything from the plate into a conical tube. I'm going to make believe this is our conical tube. Pretend we're going to pour some. We're going to pretend this is our cell media in it. So this is our conical tube with the cell media and the trypsin in it. And so next step, what you're going to do is you're going to cap it, and then you're going to go put it in a centrifuge. Now, when you use the centrifuge, you want to make sure you have a balance that is a conical tube, same size with the same volume of trypsin and media in it. But you can use water as the balance. So you want to have your trypsin and media on one side and the water balance completely opposite. Make sure you use a swing bucket centrifuge, because otherwise the cells will lyse and stick to the plate. So you're going to put this tube in a swing bucket centrifuge. You want to have it spin at 100 Gs for 5 minutes. Depending on your centrifuge, you'll have to figure out what setting that is. But it should be around setting 4 or 5 for a normal swing bucket centrifuge. Once it's spun down, you pull it out. You should be able to see kind of like a reddish liquid on top, and you'll find a pellet at the bottom. It'll be like a white pellet. Depending on how many cells you started with, that pellet may be pretty big, or it might be pretty small. So what you do once you've found the pellet, you're going to bring it back here. You're going to remove all of the media and the trypsin from the thing. Make sure not to disturb the pellet. You're going to squirt it into your waste beaker like so. You're going to keep doing that. You're going to remove the media and the trypsin, squirt it into the waste beaker until all of it's gone. If you want to be a little bit careful, you can leave just a little bit of media and trypsin on the top with the pellet underneath if you really want to be careful not to disturb the pellet. And remember to discard your tip. So now in your conical tube, you should have like a pellet, maybe a little bit of trypsin media. That's okay. The next thing you want to add is your PBS. You're going to add 1 mil of PBS to the tube, just with a pipette. So you put 1 mil. What you're going to do is you're going to take this tube, cap it, take it to a vortex, and what the vortex does, you press down, and it should like vibrate. And what that does, it'll dislodge the pellet and resuspend the cells in the solution. So when you're done vortexing, the solution should be a little bit cloudy. That's perfect. And then you're going to do the same thing again. You're going to go put it in the centrifuge. You're going to make sure you have a balance in a conical tube with water. The water level should be about 1 mil. You put it opposite your tube. Spin it in the centrifuge, 100 Gs, 5 minutes. If it's too much, the cells will lyse. And when you pull it out again, you should again have a layer of liquid here and the cells at the bottom. You're going to remove that 1 mil of PBS. It's okay if a little bit stays behind. And then what you're going to do is you're going to have your media here, and you're going to take 1 mil of media and you're going to put it in the tube. Vortex again. So you're going to have cells, 1 mil of media all together. Cells should be resuspended. The solution should be cloudy. So then comes the plating. We're going to pretend we have 2 plates here. This is 1 plate, this is another plate. Depending on your plate, that basically dictates how much media you're going to add. For our plates, we put 10 mils of media. So you're going to preferably use a stereological pipette. Take 10 mils of media, put it in 1 plate, 10 mils of media, put it in the other plate. You have 1 mil of cells suspended solution. If you're going to split 50-50, take 500 microliters of the cells solution, put it in 1 plate, 500 microliters of second cell solution, put it in another plate, swirl the plates around to make sure the cells are evenly distributed. Make sure the plates are labeled with your name, the date, what kind of cells they are. And when you're done, take the 2 plates, put them in the incubator at 37 degrees Celsius. You just watched Culturing and Splitting Cells by Hickory School's Independent Research Team. This is our prostate metastasis group. Thanks for watching.